Thank you for the kind introduction. I would like to introduce our collaboration, or the data from our collaboration with SCIEX about this new, highly versatile, high-resolution, accurate mass spectrometer for proteomics. And before that, I briefly want to explain what our lab is doing, or what we are interested in. So we have kind of four focus points of our research. One is mass spec technology, so involving new technological innovations like the 8600. The other one is clinical proteomics, so applying new technology to get biological insights. And the third one is PTM analysis to study signal transductions and gain further insights for basic research. And in order to support all of this, we are also quite active in bioinformatics, so making new software solutions in order to handle the challenges that pop up from the MS technology and applying it to clinical proteomics and mainly PTM analysis. So with this instrument, we had a couple of objectives and aims to find out what it can do. In general, we want to know if this instrument can be, or we wanted to know if this instrument can be a robust workhorse for our state-of-the-art proteomics lab. And in order to do that, we went over it with a pretty basic benchmark using a commercial K562 digest. And we were particularly interested in high-throughput workflows because we went to shorter and shorter gradients in the recent time. Of course, this is mainly interested at the bulk level, but we're also doing a lot of low-input experiments for PTMs and also single cells. And we also have a classic benchmark with multi-species containing human yeast and E. coli that's also commonly used on other instruments similar available on the market. Then one thing we were interested in is how well this instrument performs for phosphoproteomics as a benchmark for PTMs. And lastly, we want to apply our software, mainly alpha DAA and transfer learning, to get the most out of this. And I'll give a brief outlook at the end for this at my talk. So as Jose already introduced, this instrument has a lot of new features, and it basically has much better ion transfer and a lot more sensitivity than the previous generation. And this is mainly due to the new front-end optics that increase transmission. It's still like the 7600 has the ZenoTrap and EAD cell with its benefits. So thanks to the ZenoTrap, there's a high gain in MSMS sensitivity. And together with EAD, this is a very powerful combination, in particular for PTMs. So to handle this higher ion load, the TOF accelerator optics are also better and more robust than before on the 7600. And the most important change, in my opinion, is this optical detector. It basically allows the instrument to handle this higher ion load that's 10 times more than before, and can go up to 100 million counts per second, which extends the dynamic range needed for proteomics experiments. So why are these developments in particular important? So now there's only one ion source, so it's very easy to handle all kinds of flow regimes on a single setup. The only thing that might have to be changed is a probe. And thanks to this dual spray setup, we can benefit from the E-lens effect at all times, which gives us a further improve in sensitivity. I already mentioned it, but the front ion optics are now much more powerful, allowing higher transmission of ions. And in order to avoid the instrument getting contaminated 10 times faster as well, there's now a mask guard, so the instrument stays clean and with minimum downtime so far. So in the time I've been using it, I've not had to do any kind of cleaning-related maintenance so far. And of course, the highlight there, the optical ion detection, which is necessary to get good CT scan DAA data and also good EAD data. So the first thing I wanted to confirm is the sensitivity of the instrument. And I have to say I was very surprised and excited about this. For the setup, we use the EvoSep 1 or the new innovation, the EvoSep Eno, so the one generation after this. And I use a pool of 30 heavy labeled HeLa peptides for this in Zeno-MM HR mode, and this is just to check sensitivity and dynamic range over a pretty wide concentration range that kind of covers what you would expect in proteomics, so from 100 femtomoles to 0.01 attomole. So I didn't expect it to see it at the lower concentrations, but I was quite surprised. If you look here at also how linear it is in the linear view, and here also in the log scale, you can see that for at least 6 orders of magnitude, you get a very linear correlation. And you can see that you can actually detect some peptides down below 1 attomole. And you can also quantify them below 1 attomole, even without having to use the super sensitive Whisper nanoflow setup of the EvoSep. So this is kind of the more robust setup. So it can go even a bit lower at the cost of robustness if you want. The CVs were super good, below 5%. In this case, for this peptide, 2.4%. And the accuracy was also nice. So it turned out that this instrument is pretty accurate and precise if you go with a targeted analysis. So the hard specs are certainly here. And this is, of course, due to all of these innovations mentioned before. And now the exciting, or the more important question is, does this translate to a kind of discovery proteomics setup? And for this, the most important application for the future would be clinical proteomics. So we recently did an analysis of a 50,000 sample plasma cohort, and this took quite a long time. And so we went at 100 SPD for that, and we would like to go faster the next time we do something like this. And this is quite a common procedure, or it's a reoccurring thing, I would say. So within clinical proteomics or clinical related samples, the most common sample type are some kind of body fluids, so plasma, can also be urine or CSF. And these are analyzed by mass spectrometry, and from these we can gain biomarkers and then hopefully use them for precision medicine or personalized medicine. And in order to do this effectively, we need these large cohort sizes. And there were a lot of recent improvements for body fluid workflows, such as removal of highly abundant proteins, better scan modes. So this also includes scanning quadrupoles like the CT scan DIA on the 8600, and also to some part on the 7600 earlier. And the question was, how well can this instrument do it now that it has these new specs? And in order to test this, I tried the fastest gradient the EvoSep has to offer currently, which is 500 SPD. And I ran through the entire range of loads, so from 250 picograms to 200 nanograms, and ran the same methods in triplicates and compared this to what we saw earlier on the 7600. I have to say, if you look at the earlier data from the 7600 plus, for 500 SPD with CT scan, we got a really big boost. So it was a 50% boost. It went from normal SWATH from 2,000 protein groups to 3,000. But for like a deeper proteome, it was still kind of necessary, even with CT scan DA, to go to 200 SPD. But I was super happy to see that on the 8600, this is no longer the case. So across the entire dilution series, we kind of broke the records of the previous instrument. So even with 250 picograms, it was already good as normal SWATH on the 7600. And it went all the way up to approximately 5,000 protein groups for 500 SPD. So the same results, or even better results than on 200 SPD on the previous instrument, which made me pretty convinced that this is a good instrument for large-scale studies, not just also for clinical proteomics, but some basic research, such as interactomics and pull-downs, look pretty good on this instrument. And this is possible due to all of these innovations, plus the CT scan. These are kind of essential components for this. So I would like to briefly explain why this is the case. in case some of you don't know. So a normal SWATH DA, also DA in general, usually you have several fixed quadrupole windows that are based on the proteomic precursor density. And between each of these individual MS2 scan events, you will need to wait for a little bit for the collision cell to be emptied and the electronics to settle, so you have overhead time per scan. And usually also have a window overlap to handle edge effects for the quadrupole and to not mess up the quantification. And if you run it like this on the 8600, you can run with up to 133 Hertz. You have approximately 5 Thomson windows per second. And if you compare this to ZT scan, you don't have these overhead issues anymore. You have a continuous scan acquisition, and you basically save a lot of time. What this means effectively is that you can run now at 640 Hertz and have a much smaller effective window of 1 Thomson, which not only gives you more speed or more efficient quantification, but also a higher selectivity. And most importantly, from this additional Q1 scanning dimension, you have more selectivity, and if you want to, you can deconvolute the DIA data into DDA-like spectra at a virtual isolation window that's far lower than what you can get, so a lot lower than 1 Thomson, so more selective than DDA, you could say. And if you look at the quantification for this data that I showed earlier, this really shows off. So if you look at ZenoTOF, at 250 picograms, the median CV for protein groups is around 10%. So that's also pretty good for this low load. But if you go through the entire range, it stays around 10%, 10 to 7%. And this becomes a bit better with CT scan. So it's around 8% at the lower loads, and it goes all the way to 6.5% at higher loads. So it might not seem that it's that much, but it really counts for these lower abundant proteins and precursor. And concluding, I can say about this that even at super high throughputs and fast 2.2 minute gradients, you can quantify most proteins with a CV below 20%, so usually more than 85%, making this a good option for all of the high throughput applications we have planned in the future. So now the question that now that we've answered the question about throughput, what about depth? So going to very long gradients of 60 minutes and checking these mixed species experiments, we get an amazing coverage. So at 5 nanograms, it's around 8,000 protein groups and 82,000 precursors, and increasing the load to saturation, this goes all the way to 12,000 protein groups and 278,000 precursors. So this was processed with peaks. If you use Diane, the results might differ, but they're not too different on the protein group level. And it's not just the identification and quantification, it's also the reproducibility, it's rather the quantitative accuracy that's also excellent. So here on these ratios, they are pretty much nailed perfectly. There's only small deviations that can be mainly attributed also to pipetting inaccuracies. So in conclusion, for label-free quantitative analysis, you can get very good CVs and also good accuracy with a good depth, albeit you have to go to longer gradients to fully see this effect. So now that mixed species look nice, the question is how much can we get for a single organism, so a single human proteome. For this we went a bit longer in gradient time to really push it to the maximum, or rather to see what kind of the limit of the instrument is. So in a 90-minute gradient with 50 nanograms, you get almost 10,000 protein groups, and if you go up to 250 nanograms, you easily can exceed it, and you get a really nice number of precursors as well. And this also shows very well in the quantitative precision. So the CVs are below 5%. Usually I'm happy if it's below 10%, and if it's below 5%, it's really excellent. And here it's been good across the board, and the higher in load you go, or rather, if you're at medium loads, usually the CVs are the best. So in conclusion, 10,000 protein groups can identify it from triplicate injection, and 93% of them have good CVs, so almost all of them can be quantified really nicely. And yeah, we can confidently cover a large portion of the proteome, especially in nanoflow setup. So since these results looked so good, I wanted to know how well it performs with single cells and smaller dilutions. So the initial K562 single cell equivalent dilutions had super good results, so 5,000 protein groups, and if you had a kind of 2 single cell equivalent, it's even a bit more. So the HeLa cells I tried were a bit lower, but still good, so up to 3,600 protein groups from a single HeLa cell, and if you take 10 cells, you get above 6,000 protein groups. And quantification here is still good. Of course, the CVs are not as nice because single cells can be in different cell cycles, and this was just 12 single cells. So it was really just a preliminary check to see if single cells is a thing on this instrument, and I have to say you can definitely do it and get good results with a decent coverage. So now that the normal proteomes are kind of covered, I'd like to briefly go over phosphoproteomics. So this is more like a preview of how this instrument performs compared to others. So this is just one type of condition. It's 5 micrograms of HeLa digest that's enriched for phosphopeptides, injected in a quadruplicate on the Whisper-40 gradient, and the mass range is covering not the entire phospho space, so it just goes from 400 to 900 mass over charge, which is kind of the default proteome mass range. But nonetheless, the IDs here are pretty good, so there's almost 26,000 phosphosites, and 15,000 of those are class 1 phosphosites. Usually we get around 20 to 30,000 phosphosites, so this instrument performs really well, even though it doesn't even cover the complete phosphopeptide mass range. So the question is how much more information we can get with this, and since this instrument has EAD, it's very well set up to explore this in detail. We've just started doing this, so I'd like to briefly explain why EAD is good in doing PTM analysis. So this electron-activated dissociation is basically tunable ECD. So you can decide what kinetic energy you want to give to your electrons, and you can determine, or you can specify how much you fragment any kind of molecule. And this kind of fragmentation is really fast, so it can be done in less than 5 milliseconds, or typically 5 milliseconds works really well as a reaction time, and it's reagent-free, and you get the different selectivity than you would get with CID. And yeah, if you combine this with this new sensitivity, we have kind of 10 times more signal intensity than on the previous generation, which enables us to do a lot with this. And I just want to highlight, if you go for phosphopeptides with this, if you have CID spectra, you very often have neutral losses and other kinds of fragmentations that might inhibit your ability to confidently localize phosphosites or get diagnostic ions important to elucidate the structure. With EAD, you have much less of this problem. So here in this EAD spectrum for this phosphopeptide, there's almost no phosphate losses at all. But as you can see, the fragmentation efficiency is intrinsically lower, which is not a problem for most cases, but for some cases the sensitivity might not be enough. So these two fragmentation techniques have to work together in the end to get the most out of this. And I'm sure that we will do a lot of things with this in the future, especially for labile PTMs, where we saw that Without EAD, you can't even get the fragments necessary to sequence the peptide or the PTM. And in order to kind of be able to handle this also in a DIA mode in the near future, we are having our own software alpha DIA. The cool thing about this is that we introduced end-to-end transfer learning for proteomics kind of within this software package. So rather than just having some DDA data set that is used to train a model and use that on our DIA data, we take the differences we observe between high confidence matches and our library prediction and retrain this model and then search it again. And this is independent of fragmentation mechanism and instrument vendor. And I just want to show a brief example. So here on the left, you can see a dimethylated peptide before transfer learning that looks very different from what is expected, but still is a nice confident match. And after the transfer learning, you can see that we basically almost perfectly predict the fragment distribution, unlike before. But this just doesn't just apply to fragment spectra, it also applies to retention time prediction. So if you go through the transfer learning, the 95th percentile error goes from around 10% to a bit, around 3%, which gives you in the end, after transfer learning, a super good correlation between predicted and observed retention times. And effectively, this means that we can go from retention time accuracy from above half a minute to below 10 seconds, which gives us an additional boost in confidence even for very low intensity precursors. So this is not ready yet for ZT scan and EAD, but we're actively working on this, and I think this is a most efficient way to make use of the data that's to come. So with that, I would like to give you a little summary and a conclusion. So this instrument is super versatile thanks to the EAD and its CT scan DIA mode, as well as its new specs with a higher sensitivity in the optical detector. And it's very good for high throughput. It can achieve high depths at longer gradients. It can do single cells or a couple of single cells. And you can analyze PTMs very well with EAD. And with that, I would like to thank all of the people involved in this, so people from the MAN lab, as well as from SCIEX. And I'll happily take some questions now.